Hi, I usually speak a bit quiet, so if you can't hear me at the back, please do speak up. I'm going to be talking about an ever-growing chapter of my PhD. As we all know, dinosaurs to birds, in a really very reductive sense, is about the evolution of a wing. But what does that really mean for birds? Well, in manipulating the environment sense, the wings are essentially useless. So how do birds go about doing all these wonderful things that we take for granted using our hands? What appendage do they use? Well, they use the beak and the head as a surrogate hand, and if we extend this analogy further, we actually see birds using the neck as kind of like an arm, essentially. So birds get themselves into a lot of predicaments. They're large and terrestrial, they're small and terrestrial, they're scavengers, they're active hunters, they're filter feeders, burst-adapted flyers, soarers, and penguins. So how does this extra evolutionary pressure exact upon the neck? Well, one quantifiable way of looking at this is the number of cervicals. This ostrich has 19, but we can go down to 14 and even lower in chickens and passerines, and all the way up to 22 to 25 in things like geese and swans. Now we can compare this to things like mammals, which only have 7 cervicals despite neck length. And something really interesting is going on here, which we really have to investigate, but unfortunately, cervical count isn't the way to do it because it doesn't link nicely to things like genetic and developmental indicators or functional inferences. So how do we investigate this variation? Well, if we look at the spine as a whole, it's regionalised. Different vertebrae offer different functions in different regions, and we find this actually on a smaller scale in the avian neck. We find 3 regions: anterior, middle and posterior. This has been around since around 1929, and it's largely dogma at this point. People kind of take it for granted, but more recently, with more quantitative studies, we've seen perhaps up to 5, 6 or 7 regions been announced within the avian neck. Now, if this can vary, this is something we don't see anywhere else pretty much in animals, vertebrates today. Or if it is stable, we need to find and nail down this number before we can go and look into how the avian neck elongates and how archosaur necks elongate, because as we all know, if we look at the fossil record of archosaurs, necks get pretty long. So a couple of hypotheses based on this: the number of subregions in the avian neck can vary, and this number is stable, and it's the size of each subregion which varies. How do we go about testing this? Well, we use an established method by Christine Boehmer and colleagues using geometric morphometrics. Now, I know this is a shockingly low number of landmarks. That's because cervicals aren't the greatest to use in the geometric morphometrics because they change a lot across the cervical column. So to account this, we use a discrete character matrix. We combine it in PAST and produce a principal coordinates analysis as well as a cluster analysis. Now, just to take you through this data, here's our friendly ostrich again. We've got cluster analysis on the right, and we've got principal coordinates analysis over here. As you can start to see, we've got 5 regions in this ostrich. We've got atlas axis complex, we've got region 2 with just 1 cervical, we've got a large middle region, and then 2 smaller posterior regions. So 5 regions. Now, how does this extend into other species? Well, if we take these 2 cheeky little chappies, they're very different in ecology, they're very different in flight style and diet. Fortunately for this presentation, they've both got 13 cervicals, and they've both got this first atlas and axis complex, which obviously it supports the head as a whole, so this is a very functionally limited result we're getting. We see this in all the birds we study, which is close to 30 at this point, but for the rest of it, we see 5 regions. So the number is stable, it doesn't change throughout all of our birds, but what is changing is the number of cervicals for each region. Now, this isn't exactly so we can really reject this first hypothesis and start to look into how the number of cervicals within each region is changing. And this isn't that surprising considering the study that I based this on found similar results. Christine Boehmer also looked at genetic data and found it was actually Hox genes that control the subregionalisation of the avian column, and it is responsible for these 5 regions. So potentially if we extend this study to look at all these other species of birds, we can maybe look at the ancestral condition for neck elongation in Archosauria, which could be very interesting indeed. Before that, as Carl said, we've got to really dig down into the actual living things today. So these three new hypotheses emerged that external factors such as ecology and diet determines the number of cervical vertebrae per subregion, or is it like an internal factor, like a general biomechanics kind of theme going on, where it's just total neck length determines the number of cervical vertebrae per subregion? So how do we go about doing this? We make very messy graphs such as this one, where we plot subregion versus the number of cervicals per region. We get a general pattern where we have low counts at either end and higher counts in the middle. But what we need to do to assess these hypotheses now is to group birds by their flight style as a proxy for ecology or their diet. So for example, if we take a hypothetical situation such as a group of carnivorous birds, and if, for example, subregion 2 is associated with carnivory, we perhaps expect all these birds in this group to have an expansion in region 2 rather than region 3. Do we get this? No, we don't. It's a very messy situation, even in the flyers and in the diets. We do get shifts from this general pattern, but we don't get a clear shift in this pattern, which kind of leads me to the conclusion that these aren't predictors of subregion makeup. But we get a really interesting result when we plot neck length normalized for body mass over the number of cervicals. Regions 1 and 5, so these terminal regions in the neck which support the head and the head and neck as a whole, remain stable across neck length in all birds. But region 3 decreases with increasing neck length, and regions 2 and 4 increase with increasing neck length. So what does this mean? Well, firstly, we can accept that third hypothesis, that total neck length is the predictor in subregion makeup. But I think we're really observing general patterns in how the neck has evolved with subregionalization. It's more adapted to changes in general biomechanics, such as the neck, rather than to specific, special cases like diet or ecology. And this has been vaguely mentioned by van der Leeuw in 2001. And what I think is really cool is regions 2 and 4 seem to be the prime sites of neck elongation in birds. So what can we do with this data? Well, we can try and analyze why we see this pattern in terms of morphology and anatomy, but we can also try and see what this means for neck evolution in Archosauria. This is all work that's not really been done yet. This is more me trying to hint at what I'm going to do next. But if we look at the morphology and anatomy of cervical vertebra, now this is for a penguin, but this pattern applies broadly across most of our species of birds that I've studied. We take one feature of anatomy, the neurospine. We see that it's large in regions 1 and 2, decreases in region 3, and then increases again in region 4 and 5. Now notice region 2 and regions 4, these sites responsible for neck elongation. The cervical rib, the neural spine is larger. Now what's the neural spine for? Neural spine is attachment sites for dorsal musculature and dorsal ligaments, which are responsible for supporting the head and supporting the neck as a whole. So if you're getting more vertebrae in these regions, you're essentially getting more neural spines, which are allowing for more attachment sites. Now, I have to, as a caveat, admit I've not gone in and dissected this just yet, but I'm going to try and quantify this myself later on in my PhD. But what I find super interesting at the moment, we can start to glimpse at what Hox genes might be associated, and that's a big might, with neck elongation in archosaurs. So we've got this new region, region 4, in birds. We've also got region 2 as a site which is associated with neck elongation. The ancestral condition is 2 cervicals. But we also see region 2 is associated with neck elongation in the only piece of work that's been done on sauropods, Palaeosaurus. So what I'm trying to maybe hint at here, that future work should really focus on region 2 as like an ancestral site for neck elongation, and potentially archosaurs, and if I'm really putting it out there, maybe even sauropods. And finally, I think it's really cool where this region 4 can potentially evolve, and that's another potential future work that I really want to get into at some point. So to conclude, we can really do away with this dogma that's been around since 1929. We've got 5 regions, or 3 in all birds. Regions 2 and 4 seem to be responsible for neck elongation, and all the necks, we seem to be changing in neck length. The regions are associated with that rather than external factors such as diet or ecology. And like I said, this region 2 is really important, potentially as an ancestral site of neck elongation, which is hopefully going to be some really cool work coming out in the next 2 years or something. So I'd like to thank supervisory team, scanning, funders, and you for listening, and I'd happily take any questions. Thank you.